is here to tell you, I have not heard this talk, so
No, this is new.
Yeah, so, not that I hear all, I mean, not that we should have already heard talks that are coming, but this sounds interesting, and Bandana's going to speak to us on the role of nuclear receptors in drug metabolism. So thanks for coming all the way from the great IBT.
This was good. I really enjoyed. So as Mike was saying, that this is actually a relatively very new type of work that we have been involved in our lab. And basically my lab focuses on studying nuclear receptors, their role, both steroid and non-steroid receptors, their role in drug metabolism, steroid and drug metabolism, and also their role in hormonal cancer, specifically androgen-induced prostate cancer. But today I'm going to share with you some of the published and unpublished data. We are in the process of writing up some of the unpublished data, where we actually look at 3 different nuclear receptors that can sense drugs and xenobiotics as they come to the GI system and the liver for metabolism. And then, nuclear receptors, they are transcription factors, and then how they actually are involved in drug and steroid metabolism. So, nuclear receptors, it's a large family of ligand-inducible transcription factors, and all nuclear receptors, there are 48 different members currently known in the human. All nuclear receptors, they have the same type of overall structure with distinct functional motifs, such as the DNA binding domain. This is the domain that directly interacts with the hormone response element in the target gene, so that receptor binds to the target gene to elicit its transcriptional regulatory function. And this is the ligand binding domain, where the ligand, such as for steroid receptor, it would, like androgen receptor, it would be androgen, or for estrogen receptor, it will be estrogen. So it's a ligand binding domain that interacts with the hormone itself. And so all 48 members of nuclear receptor superfamily would have this overall structure. And the DNA binding domain of this structure is highly conserved. It's very, very conserved within a particular subfamily. For example, if you look at steroid receptors, that is androgen receptor, estrogen receptor, progesterone receptor, glucocorticoid receptor, mineralocorticoid receptor, they have extremely conserved sequence within the DNA binding domain. But if you go outside the subfamily, but within the nuclear receptor superfamily, you will still see considerable amount of sequence conservation within the DNA binding domain. And then the ligand binding domain is less conserved than DNA binding domain, but still pretty good conserved. And then there is this N-terminal domain, which is highly variable even within a subfamily. For example, the N-terminal domain of androgen receptor, even for human and rat, it would be quite variable. And then these are the activation functions through these domains, the transcription activation function of different nuclear receptors, they are carried out. Now today, as my title suggested, that I'm going to look at how nuclear receptors are involved in drug metabolism. And
do we have questions?
Sure. Yeah, please.
I won't interrupt, but often. But does that mean that the specificity for receptors lies in the in that AF1,
Not
the
really
N-terminus?
Well, not really. For example,
Specificity
if you no, not really. First of all, even the DNA binding domain is highly conserved, but let's say within a subfamily such as steroid receptor, but there is enough 1 or 2 amino acid difference that is going to determine target gene specificity. That's number 1. Number 2 is that within the target gene, so here is the receptor that's eventually going to bind to the target gene. But within the target gene, there are adjacent sequences where other transcription factors would bind, and the interaction between those non-receptor transcription factors with receptors would also would also be a determinant for specificity of which target gene would get activated. And then it's more complex actually. It's the co-regulatory proteins that interact both with the N-terminal domain and LBD that is going to also determine specificity. Okay, so today I'm going to concentrate on a special type of nuclear receptor and there are two of them. And these are xenobiotic sensing nuclear receptors, and they were discovered about 6 to 7 years ago by Ron Evans and his group, and also by David Moore and his group. Ron Evans is in Salk Institute, and David Moore is in Baylor College of Medicine now. And so these are called PXR, standing for pregnant X receptor, and CAR, standing for constitutive androsten receptor. And what these drugs do, these receptors, so again, they are member of the nuclear receptor superfamily, so they again would have this kind of overall structure. But they bind to the target gene. As a matter of fact, all non-steroid receptors, including PXR and CAR, would bind to target gene as a heterodimer with retinoid X receptor alpha. So RXR, which itself binds to the 9-cis-retinoic acid, as opposed to all trans-retinoic acid, that's the ligand for RAR, or retinoic acid receptor. But RXR is really a master nuclear receptor because this kind of heterodimerization is an obligatory feature for all of these receptors to work. And here I show you the example of PXR and CAR, the two xenobiotic receptors, because all of you realize that drugs are also xenobiotic compounds. All the medicinal drugs that we take all the time, they are foreign compounds, they are synthetic compounds. But even for thyroid hormone receptor or vitamin D receptor or retinoic acid receptor, all of those receptors would also have to heterodimerize with RXR, the master receptor, in order for them to bind to the target gene. Now, talking about steroid and drug metabolism, as we all recognize that there is this phase 1 step in the metabolism, which is really the oxidation step carried out by monooxygenases, specifically this whole superfamily of cytochrome P450, and very commonly known as CYPs, or the CYP enzyme. So that's the first step, usually, where the drug or the xenobiotic compound and even some endogenous compounds such as bile acids and hydroxysteroids, and you'll see some examples of that later, they would get oxidized by these heme-containing monooxygenases. And that hydroxyl group usually is not enough. Then there is a second step that goes on, which is carried out by phase 2 enzymes. And the examples will be glutathione S transferases, GST-pi and other members. And also UGTs, another type of uridine glutathione transferases. But the one that I'm going to be concentrating on are again a superfamily of enzyme known as SALTs, that stands for sulfotransferases. And we have been working with sulfotransferases for many years. As a matter of fact, I started out my academic career working with sulfotransferase in a very different context when I was studying gene regulation during aging. And that's when we actually discovered one of the SALT enzyme being a senescence marker protein. But now, after all these years, it has beautifully come into an overall physiological picture in terms of being a phase 2 enzyme and playing a very important role in drug and steroid metabolism. And also in this metabolic regulation, there is a phase 3 step that's carried out by the transporter proteins, because one has to realize once the drug and xenobiotic compounds taken up by the system comes to the GI system and to the liver, then they get metabolized by phase 1 and phase 2 enzymes, but then they have to be transported out. First they have to be transported in from the intestinal lumen, and that is carried out, that is with the help of these transporter proteins such as multi-drug resistance. There are many of them. I just gave a couple of them. And then MRP2, MRP1, and all these other transporter proteins. And then, once they get in with the help of these transporter proteins, they get metabolized. And then those metabolized products would have to be then effluxed out of the cells into the intestinal lumen for ultimate clearance. So this phase 3 step is also very important in this metabolic regulation. Now it turns out that these two nuclear receptors that I just talked about, PXR and CAR, these xenobiotic sensing receptors, they play central roles in the induction of the enzymes that code for this cytochrome P450 and all these phase 2 enzymes, including the SALT that we study, and also the transporter proteins. So that's how these two receptors are, and they get activated when these drugs and there are some endogenous steroids also can activate these receptors. So these steroid drug or other xenobiotic compounds, they actually bind to PXR and CAR and activate, just like a estrogen receptor would get activated when estrogen would bind to estrogen receptor. So these receptors, when they get bound to their ligand, and for them the ligands are all these compounds, they get activated, and then they can transcriptionally induce all of these players in xenobiotic and drug metabolism. So just to give you some example how diverse the ligand profile is for PXR and CAR, so PCN, it's an anti-glucocorticoid. It's a very strong inducer of PXR, and basically PCN binds to the ligand binding domain of PXR and then activates PXR, so that now PXR actually heterodimerizes with RXR and then binds to target gene. Rifampicin, all the any drug you can think of, Tylenol or acetaminophen or any antibiotic, anything you can think of that we are encountering all the time in our daily life, all these drugs when we take, they actually bind to PXR. And so then the puzzle is that how can compounds with such diverse structural aspects could still so specifically bind only to PXR or in some cases CAR and then activate the receptor, And the extracrystal structure of these receptors actually has provided some clues. But anyway, so even RU486, the abortion drug, that would activate PXR. Of course, glucocorticoid would activate PXR. And in the terms of CAR, phenobarbitals, they activate CAR. And this is a pesticide. So it's not just drug, the xenobiotic compound, other than drugs, therapeutic drugs could also activate this class of xenobiotic sensing receptors. Now, but the surprise came about 2 years ago when we realized that even vitamin D receptor, which is long known as a pivotal player in bone physiology through regulation of calcium and phosphate homeostasis, also could be involved in regulating drug and xenobiotic metabolism. And again, just to make the point, vitamin D receptor is a non-steroid nuclear receptor that would also have to heterodimerize with RXR, the master receptor, in order for this heterodimer to bind to the target gene at the vitamin D response element. So this was the big surprise. Okay, the first surprise was the discovery of PXR and CAR as two xenobiotic sensing members of nuclear receptors that can be activated by such diverse class of compounds. But the second surprise came about 2.5 years ago when a paper was published in Science by a group in Dallas, Mangelsdorf's lab, showing that vitamin D receptor can actually activate cytochrome P450 enzyme gene transcription. Okay, and we were also on the same track, and we were a little late in publishing, but we actually showed as a second example that vitamin D receptor can transcriptionally induce our SALT enzyme. And remember, SALT is a phase 2 enzyme and CYP is a phase 1 enzyme. So this then gave the field the examples of both phase 1 enzyme and phase 2 enzyme in the GI system and in the liver can actually be induced not only by PXR and CAR, but also by vitamin D receptors. So basically in today's talk, I'm going to concentrate on vitamin D receptor regulation of our SALT enzyme, which is SALT2A1, and I'll explain that a little better later, and how that might be physiologically, how that regulation could be physiologically a very important regulation. So again, to make it, to recap what I just said, so our enzyme is SALT2A1, which is actually, long known as DHEA sulfotransferase. But I just have to mention that this enzyme, we didn't know what it did at that time, but many years ago, when I was a postdoctoral fellow, actually, we discovered this gene as a senescence marker protein that is highly upregulated in the liver at old age. But anyway, so this sulfotransferase is a phase 2 enzyme and is transcriptionally induced not only by vitamin D receptor, but we also showed it's induced by PXR and CAR. And so originally, as I said, about 2.5 years ago, the science paper showed that VDR could upregulate CYP3A gene expression. Now you have to realize CYP3A family members, especially CYP4A, 80% of the drugs that we take, that they get metabolized by CYP4A. And CYP3A is also actually it's a CYP3-4A or something, this member. So it's a very, very important metabolizing enzyme in the context of drug pharmacology. And actually, it's, yeah.
CYP19, is that the aromatase?
Yeah.
Is that affected by these? Is that
No, it's not. Usually it's because it's aromatase is the steroid metabolizing enzyme. I don't think anybody has looked at it, but mostly for this in the CYP field, it's the drug metabolizing CYP3-4A and
so CYP19 is a different.
It's still because remember the CYP family is a super family, and all these steroid metabolizing enzyme, not only aromatase, but all the other hydroxylases that are involved in the steroid biosynthesis pathway, they are also member of the CYP family. But as a matter of fact, it would be interesting if you have the aromatase gene, we could actually look at whether or not, because nobody has looked at it. I've often thought about it because this, since we are set up for it, it's very simple to check that, and if it works, I think this would be new finding. So we could talk about that. But anyway, but the thing that that I got excited about when I found that the SALT2A, this DHEA sulfotransferase, could not only be activated by PXR and CAR in its transcriptional process, but also by vitamin D receptor. So just to talk a little bit about DHEA sulfotransferase, it belongs to the SALT family of sulfotransferases, and there are actually 5 subfamilies, SALT1 through SALT5, and SALT2A1, as the number suggests, is a member of the SALT2 subfamily. Basically, SALT2 subfamily so far is quite small. It's SALT2A1, which is the original DHEA sulfotransferase. The name came because the DHEA is a very good substrate for this enzyme. But this enzyme also would act on testosterone, estrogen, pregnenolone, and dihydrotestosterone. It's also a very good bile acid sulfotransferase, and I will come back to that later. As a matter of fact, SALT2A is exclusively the enzyme that is involved in the sulfation of bile acid, and as I will discuss later, lithocholic acid, which is a very important byproduct from primary bile acids like cholic acid and chenodeoxycholic acid. Lithocholic acid, if it doesn't get cleared out efficiently by the body, from the body, it's very toxic. And this way, sulfation, once the lithocholic acid gets sulfated, because it's highly polar, it gets very easily cleared out from the body. Now I'll come back to this later. So basically then, so SALT2 family is kind of short because SALT1 is very big family and they work on aromatic compounds, but SALT2 it's also known as hydroxysteroid sulfotransferase. It's also known as bile acid sulfotransferase. because of the substrates that they act on. But there is another member which we have become very involved in studying, is SALT2B. And it's so interesting that SALT2A is expressed in the enterohepatic tissues, liver and intestine. It's a little bit expressed in the brain, but that's about it. But SALT2B is not expressed in the liver at all, but it's highly expressed in the prostate. And because one of our other projects involves androgen action in the prostate, we have become very active in understanding. And again, SALT2B is a very newly discovered member of the family. It got discovered about three years ago. But since it's expressed in the SALT2B is expressed in the prostate, we are studying its role in the prostate in metabolizing androgenic steroids. Okay, and so as I have already discussed, that SALT2A is involved in the inactivation, detoxification, and clearance of hormones, because all of these are endogenous hormones, and also drugs, toxins, and other xenobiotic. And I have mentioned that we got involved in SALT2A1 because this was really first identified in our lab when I was a postdoc as a senescence marker protein. Very quickly, a group in Research Triangle Park, they have produced the extracrystal structure of SALT2A1. The monomer structure is like that, but the SALT2A1 acts as a dimer, homodimer, where the crystal structure looks like that. And this is the enzymology of the enzyme. So it will act on the 3-hydroxyl group of the steroid structure, hydroxysteroids such as dehydroepiandrosterone, sex steroids like androgen, estrogen, and bile acids, because bile acid has overall core structure like that. But the interesting thing is that this enzyme will also act on drugs such as budesonide, which is an anti-inflammatory drug. It also acts on tamoxifen. We all know what tamoxifen does, anti-estrogen, etc. So, but it's a simple enzymatic reaction where it uses PAPs. This is the structure of PAPs, and it uses PAPs as a cofactor. So it gets the sulfate group from PAPs, and that sulfate group gets attached to this hydroxyl group to form the sulfated metabolite, in this case DHEA sulfate. So this is a way we actually assay for this enzyme activity in whichever tissues we want to measure. So the physiological significance then of SALT2A1 activity would be, as I have mentioned, in steroid, drug, and xenobiotic metabolism, in the maintenance of cholesterol homeostasis, because remember, cholesterol breakdown in the liver produces the primary bile acids, cholic acid and chenodeoxycholic acid, and so the homeostatic balance between cholesterol and bile acid is very important to maintain cholesterol homeostasis. And we actually have shown in another published paper that the bile acid receptor, which is another member of nuclear receptor superfamily known as FXR, also upregulates this sulfotransferase. Now, physiologically, it's important to understand that this sulfotransferase is involved in inactivating androgenic hormone, because sulfated androgen, they are receptor inactive. So that's how they could be, or sulfated estrogen would not be able to activate estrogen receptor. That's how these sulfotransferase would have preventive role in androgen, actually I should have said hormone-induced cancer, such as in breast cancer through estrogen metabolism, and in testicular cancer through androgen metabolism. Very quickly, this is a result from our lab to show that PXR is activated by PCN. So in mouse, if you inject mouse with PCN, a PXR activator, there'll be induction of SALT2A1. This is a northern, and also there is induction of CYP3A. And here is the data where vitamin D receptor is, this is the one that showed that vitamin D receptor is an inducer of SALT2A1. And this is a cell culture experiment where the cells were treated with vitamin D or vehicle, and then we looked at the SALT2A1 messenger RNA level. This is the endogenous level, and there is a very strong induction. We also showed it in the mouse when mice were injected with vitamin D or vehicle, which would be ethanol, there is induction of SALT2A1 shown by Northern. And we actually have Western blot data of this to show this increase in messenger RNA reflects increased amount of protein. So the two data that I just showed in the two previous slides shows that SALT2A1 is activated by PXR because PCN would activate PXR, and SALT2A1 is activated by VDR. And as a matter of fact, this is in a mouse SALT2A gene. We actually have shown that there is direct it's the same regulatory element that binds either VDR or PXR. And there is actually a competition. This is a transfection as an experimental model, and we can demonstrate that this is this response element that can bind either VDR or PXR. So initially, in the cell, this experiment was done in liver cells. If you just do just VDR, if you transfect with VDR, but no PXR, no exogenously added PXR in the transfection system, so there is like about 5-fold induction in response to vitamin D. But that response steadily goes down with increasing amount of PXR added to the transfected system. So, which means that physiologically, something like SALT2A gene, that the same regulatory element could be activated either by PXR or by VDR. It's the overall milieu, the endocrine milieu within the cell that would determine whether VDR would be a predominant at a particular point, whether vitamin D would be a predominant regulator for this gene, or whether PXR would be a predominant regulator of this gene. Okay, so then we ask the question, okay, we know that all the CYP enzymes, they get activated by VDR. This is work from other people that came first, namely Mangelsdorf at Southwestern Dallas. And then we know that SALT2A is also activated by VDR. But is there any exclusive advantage in the regulation of SALT2A induction by VDR? And for that, we have to keep in mind these 2 points. First of all, in humans, LCA is metabolized almost exclusively as LCA sulfate. And because if you look at human urine, LCA is most predominantly found as sulfated LCA. And the second thing, that SALT2A is the exclusive enzyme available for sulfating LCA, lithocholic. LCA is the lithocholic acid. Just quickly, cholesterol metabolism pathway. So in the liver, cholesterol gets broken down to chenodeoxycholic acid and cholic acid. These are the primary bile acids, and these primary bile acid in the intestine through the intestinal flora gets converted to secondary bile acid, which is the lithocholic acid LCA and deoxycholic acid DCA. Now, the secondary bile acid, and especially lithocholic acid, as you all probably know already, that is extremely toxic if it accumulates in the cell. So therefore, sulfation of lithocholic acid exclusively by SALT2A1 is a very good protective mechanism to get rid of lithocholic acid. There you can look at old literature and you can actually find evidence for LCA-induced pathology. For example, LCA induces DNA damage and promotes cellular hyperplasia. There are published papers showing this kind of thing. Actually, LCA would form DNA ducts. An in vitro experiment has shown that. And also, epidemiology shows that LCA may have a role in the etiology of colon cancer. There's an epidemiologic evidence, and also there is experimental evidence in animal models to show that LCA, if you give if you feed the animals lithocholic acid, high dose of lithocholic acid, that would lead to colonic hyperplasia of the colonic cells, and that could progress to cancer. So, and in more practical terms, there is also evidence from old literature that if you take high-fat diet, then you stimulate production of LCA. So in the Western diet, when there is high-fat diet, that could cause LCA overload, or there could be LCA overload because of some specific metabolic disorders. So either through endogenous mechanism or through exogenous mechanism, such as high-fat diet, one could envision that we could get into LCA overload, especially in our intestinal system. And because the experiments have shown that under these conditions there would be high level of LCA in the fecal excreta. And there, if then vitamin D receptor is abundantly present in the intestinal cell. And then induction of SALT2A by vitamin D receptor would be a very good protective mechanism to counteract this sort of potential hazard. So as I have been already mentioning, that since sulfoconjugation of LCA is mediated exclusively by SALT2A1, we could, because we could at this point we have evidence from molecular or cellular experiments. We don't yet have all the animal experiments done, but we could expect that SALT2A1 would protect the hepatic, biliary, and intestinal tissues from LCA toxicity. As a matter of fact, a paper came out in JBC in 2003 where they, in mice, they actually showed that SALT2A1, which they call hydroxysteroid sulfotransferase, which is same as, now the common name, the commonly accepted name is SALT2A1. But the SALT2A1 would have a protective role if you give a lot of LCA to the animal, but depending on the level of SALT2A1, that would protect the liver.
So,
Yeah.
That means there's no knockout, is it?
SALT2A1 knockout, no. But those receptor knockout, like if you have PXR knockout and then if you have FXR knockout, or if you have CAR knockout and you breed them to have double knockout, and then you cut down the SALT2A level totally. And with those models, they have shown actually that, so if you have a wild type versus a knockout, your protection is much better when you have the wild type under the same. Yeah, that sort of experiment has been done. So the thing is that we ask the question, okay, so would LCA, so we know that SALT2A is induced by vitamin D receptor. And I'm making a case that LCA, so if when we have LCA overload in our system, in our intrahepatic system, either through metabolic disorder or through high fat diet or whatever, if SALT2A, so the protective mechanism would be for induction of SALT2A1, right? Because then lithocholic acid would be taken care of as sulfated lithocholic acid, that would be rapidly eliminated. So would lithocholic acid induce SALT2A? And sure enough, it does. This is the vitamin D experiment. This is the mouse SALT2A1 and the human SALT2A1. Actually, this is published, but this is an unpublished data in our lab. We actually could show that human SALT2A, we didn't do the experiment with the mouse, but because we are already always interested in the human, especially because in human lithocholic acid sulfate is the exclusive or almost predominant metabolite of LCA. So we looked at human SALT2A. This is just a transient transfection experiment, and again we can see threefold induction by LCA acetate. But why is so? What's the molecular mechanism? Well, it turns out that again Mangelsdorf lab, in that science paper that I talked about, which came out 2002 summer, he showed that lithocholic acid is actually a ligand agonist for vitamin D receptor. Isn't that strange? But it is, So vitamin D receptor not only can be activated by its classic ligand, which is the vitamin D, but it actually can be activated by lithocholic acid. Because Mangelsdorf's paper published 2002 summer showed that LCA activated VDR can induce cytochrome P450 gene transcription. And actually this year another paper came out in molecular endocrinology. They looked at many different members of cytochrome P450, and all of them get induced by LCA. So we actually, this is an unpublished data from our lab, we actually asked the question, okay, so then what happens in the cell, in live cell, when cells are treated with LCA acetate or vitamin D? So and this is a vitamin D receptor that is tagged, fluorescently labeled with CFP, cyan fluorescence protein. So this is a CFP BDR that we transfected into the cell. This is COS cell. We also have data with the intestinal Kako cell. And this is ethanol treated cells. These are different cells, okay? In the microscopic view, so we just took pictures of different cells. You can see that, in some cells it's almost equally distributed between nucleus and cytoplasm. Some cells it's more in the nucleus, but still quite a bit in the cytoplasm. And then again, this is in between. When you do treat the cells with vitamin D, again, it's mostly goes to the nucleus. That's what you would expect. But the interesting thing is that when you treat the cells with LC acetate, the same thing happens. Vitamin D receptor gets redistributed from the cytoplasm almost totally into the nucleus. And this would make sense. So, this is just another way of showing that LCA actually would bind to vitamin D receptor, will push it to the nucleus totally, where then the activated vitamin D receptor would elicit its gene transcription function on the phase 1 and phase 2 enzymes. Now, so far, the vitamin D receptor regulation in the xenobiotic or drug pathway only shown in the case of cytochrome P450 genes and the SALT gene in our system. But I can bet that if you look at the phase 3 transporters or any of the systems that are involved, because you need all of those phase 1, phase 2, and phase 3 to work hand in hand to regulate xenobiotic metabolism. So all of them would be regulated by vitamin D receptors also.
I'm just curious, what
Yes.
kind of cell do you use?
That's a cause cell, I told you, cause cell. Because again, we have to find cells that don't have much of vitamin D receptor. We also have done this experiment in Kako cells, which is the intestinal cell, and the results are not as clear cut because of the endogenous vitamin D receptor. But the important point is that you wouldn't expect lithocholic acid, and I showed you the structure before, that would actually bind to VDR and will bring it to the nucleus, but it does. Okay, and then because we have been always very interested in understanding the molecular mechanism of of any particular gene regulation. So we did some studies, and the interesting thing that we found that in the case of human SALT2A, this VDR mediated regulation of SALT2 gene is totally dependent on CBP alpha, not CBP beta, but CBP alpha. CBP stands for CAAT, cat enhancer binding protein. It's a very liver and intestine-rich transcription factors involved in fundamental way in regulating a whole cascade of genes in the liver and intestine. So what we did, first of all, we took the human SALT2A promoter and did deletion studies, and in transfection model looked at the vitamin D induction, and we see that vitamin D response region lies somewhere between -211 and -127 position. So we looked at the sequence of human SALT2A promoter, and we see lots of sequences that are, this should be here, I was playing with this slide, but somewhere here it went up. But anyway, the thing is that, you see we marked all these different boxes. Those are all potential nuclear receptor binding site. It turns out that, in the case of steroid nuclear receptor, the motif that binds to the receptor is kind of fixed. It's TGTTCT and 3 base spacing, and then again complementary sequence. But in the case of non-steroid receptors, including vitamin D receptor, PXR, and CAR, it could be DR0, it could be DR4, DR1, DR5, DR6, and in the case of SALT2B, we just discovered it's DR7. And that's one of the Endocrine Society poster. We are in the process of writing up the paper also. So what we did, since our deletion study showed that the response region is somewhere between -2 something and -127, that area we did footprinting with liver nuclear extract. We actually found within that response region 2 footprints, and we call that footprint 1 and footprint 2. And by different kind of experiment, we actually could prove that footprint 1 is a CBP binding site. Now, it could be because both CBP alpha and CBP beta would bind to the same element, and then footprint 2 is actually vitamin D receptor binding site. Well, this is just one such experiment that would show that, but just for time, I'm not going to explain. But this is more interesting. So what we did, we took the promoter. Here is our footprint 1 site. Here is our footprint 2 site. We either point mutated footprint 1 or point mutated footprint 2. These mutations, again, we demonstrated that these mutations would cause loss of binding of CBP alpha or beta to footprint 1 and vitamin D receptor to footprint 2. And you can very easily see that the wild type promoter showed the vitamin D dependent induction, but if you mutate either footprint 2, which you would think, that's a vitamin D receptor binding site, that would abolish the induction. But if you do the CBP mutation site, if you mutate the site that binds to CBP, you still lose you actually could see inhibition. And again, of course, the double mutation also shows loss of induction. And we also did, because one of the goal tests in characterizing this kind of regulatory elements is whether or not they would work in a heterologous promoter context. So what we did, we took 3 copies of footprint 2, ligated to thymidine kinase, that's a basal promoter, and then luciferase. Same way, footprint 1, 3 copies of it to thymidine kinase and luciferase, or the 2 together, footprint 2, 3 copies, footprint 1, 3 copies to thymidine kinase. And as you can see, even footprint 2 by itself could not show vitamin D dependent induction. Of course, footprint 1, which is a CBP site, would not show that. But when you have two of them together, you see very nice induction. Okay, this then tells you that CBP element is needed in order for vitamin D receptor to be able to induce this gene. And but we wanted to have more. Again, we are in the process of writing up this paper. So, and reviewer gets very nasty. He always tries, he or she tries to knock it down as much as can. So, we wanted to have additional evidence that CBP plays a role. And plus, with the experiment that I just showed you, you really cannot distinguish between CBP alpha and CBP beta, because both can bind. When you do EMSA, electrophoretic gel mobility shift assay, both CBP alpha and CBP beta would bind there. So what we did first, we wanted to have a cell line that does not express CBP, okay? And you can see CACO2, that's the intestinal line, this is a liver line, HepG2, all of them express vitamin D receptor, all of them express RXR alpha. But in the NIH-3T3 cells, the CBP alpha level is, in Western blot analysis, is undetectable. On the other hand, CBP beta is still there, okay? So here we already got a system, a cell line, which does not express CBP alpha, it still expresses CBP beta. So not only it would allow us to again determine the role of CBP in this induction, but it would also allow us to distinguish between CBP alpha and CBP beta, hopefully. So what we did is that we ran such experiment, transfection experiment. If you would just concentrate first on this panel. So this is the wild type SALT2A promoter that contains both the footprint 1 and footprint 2. And you can see that if you don't have, because this is NIH-3T3 cell and they don't have CBP alpha. So if you do not co-transfect, this should come here, okay? These are minus. It just got shifted a little bit. But if you do not add CBP alpha expression plasmid into the cell, there is no vitamin D dependent induction, okay? However, if you co-transfect these cells with CBP alpha wild type, these two will be plus, and again, this got shifted. This plus corresponds to these two panel. And now you can see very nice threefold vitamin D dependent induction. On the other hand, we have a mutant CBP alpha that is not able to bind to the DNA. So when you co-transfect these cells with mutant CBP alpha, again, you don't see this induction, okay? So in the vector transfection, so this one was vector transfected, these two, So this does not show any vitamin D induction. Now you already know that NIH-3T3 cells does have CBP beta. So that already will then discriminate you between CBP alpha and CBP beta. And I don't have the data here, but we have more recent data to show that when we actually co-transfected, we could argue maybe it doesn't have enough CBP beta. You need some more boosting. So we actually, in these cells, we co-transfected the cells with CBP beta expression plasmid, but then even there is no induction. So this way you very clearly can distinguish between CBP alpha and CBP beta. So this paper that we are writing, not only we are saying that you need CBP for vitamin D dependent induction of SALT2A, but we actually can say CBP alpha, not CBP beta, is essential in the process. And you see, so this is the -127 promoter, which has lost both footprint 1 and footprint 2, and there is no induction. And interestingly, CYP3A, which is also regulated by vitamin D receptor that others have shown, but that does not need CBP, because even with the minus, these two lanes, you see very strong vitamin D induction. And this is actually a heterologous promoter that we got from somebody very kindly gave us from Vanderbilt University. And again, with the heterologous promoter that I showed before, you have 3 copy of footprint 1, 3 copy of footprint 2 to a heterologous thymidine kinase promoter. Even that one needed CBP wild type to see this induction. If you have the CBP mutant that cannot bind to the DNA, you lose this induction. So I was really happy to see this data, because initially we had this data, but that's like an element, you muted the element. But so we could show in a complementary approach by using overexpression, exogenous overexpression, that CBP alpha is needed. Yeah.
There are good antibodies out there for CBP alpha and beta,
Right.
so you could still show it by super shifts too, right?
No, yeah, but the problem is that if you just take a EMSA and if you just take an element, like a footprint 1, and if you take liver nuclear extract, you will see a very nice, actually I have this data, I can show you this data.
But CBP, if CBP beta is not binding,
No,
it shouldn't be
it will bind. In vitro it will bind. So that's the problem with the assays such as EMSA. It's an in vitro assay. It doesn't really give you the physiological
here
content.
it's-
For example,
Oh,
this is an experiment where you have antibody to, this is the complex, and when you add antibody to CBP alpha, there is super shift. When there is antibody to CBP beta, there is super shift. And you'll get that. Liver has both CBP alpha, CBP beta. It's an in vitro assay. You'll see binding of both. Okay? So that's why you have to really go to the cell to see what's happening. Okay, and another experiment we did is the CHEAP assay, chromatin immunoprecipitation assay, which basically could demonstrate that vitamin D receptor is recruited to this regulatory region. I don't have time to explain the principle of CHEAP assay, but this is a very nice assay to show, because not all transcription factors actually bind to the DNA element, but still could be regulating this gene, because there could be transcription factors that do not directly bind to the DNA element, but interacts with the DNA binding protein. So they are still recruited to the regulatory site without directly binding to the regulatory element. And we actually could show that in a time-dependent fashion that vitamin D receptor in response to this is the addition of hormone, the time, vitamin D receptor is recruited to the site, same is CBP alpha with the same kinetics is recruited to the site. And these are some of the other experiments to show that not in response to ethanol, but which is the vehicle, but in response to vitamin D, the co-regulator, the P160 co-activators, SRC1 and GRID, and also CBP, which is P30, they are recruited to that regulatory site, but not NCORE, which is a co-repressor, and you wouldn't expect that. So, and also we showed that there is physical interaction. We could demonstrate there is physical interaction between CBP and VDR. This experiment was done in the Kaku2 intestinal cell, because this kind of study you really like to use either liver cells or intestinal cells, because that's the system that we are talking about in an in vivo context. So again, co-immunoprecipitation study clearly showed that CBP, if you immunoprecipitate the cell lysate with CBP alpha antibody and then Western blot that immunoprecipitated protein with vitamin D receptor antibody, I should say, alpha here. But anyway, you see this band which corresponds to vitamin D receptor. And then you can do the reverse experiment where you immunoprecipitate with vitamin D receptor and then Western blot with CBP alpha. You still see the complex. The other interesting thing I saw that this interaction, CBP alpha VDR interaction, is DNA dependent. Because first of all, there is a test called ethidium bromide test. If these two proteins, when they interact physically, if that interaction is DNA dependent, then if you add ethidium bromide to the reaction mixture, that interaction will go away, and it did. But more importantly, if you look at this data, so this is the hormone treatment time, and again this is a immunoprecipitation with CBP alpha and Western blot with anti-VDR antibody, and we saw that there is a hormone dependent increase in that interaction that you see here. Interaction goes more at 120 minutes, but there's also a DNA dependent interaction. So if you add that we took a oligo which contains the footprint 1 and footprint 2, and that DNA, when added to the reaction mixture, incubation mixture, there is further enhancement of the interaction. So that's more or less about it. So I just like to go to the overall summary then. SALT2A1, we have been studying it since early 1980s or middle of 1980s when we cloned this gene as a senescence marker protein. It has turned out now to be a phase 2 enzyme, and the surprising result is that it's now induced by vitamin D receptor in the enterohepatic tissues, and this VDR-mediated SALT2A induction physiologically may be essential in the detoxification of LCA overload in humans. And this VDR-mediated SALT2A induction may have evolved as a defense against high-fat LCA-mediated injury. When we take high fat, that produces a lot of LCA, and if our system cannot cope with it, namely by not being able to induce enough SALT2A to make enough LCA sulfate, that would be injurious to the GI system. It's just kind of a schematic, and basically what I'm trying to say here, here is the intestinal lumen and this is the enterocyte. LCA coming, the primary bile acid in the enterocyte is becoming LCA, and then you need the induction of SALT2A, primarily by PDR, but perhaps also by PXR and CAR to make LCA sulfate. That is necessary to protect the GI system from LCA-mediated pathology. And so here is my current lab composition. And T-Sam and Chuck, Dr. Song, these are the two people. They were primary players in this VDR regulation of SALT2A. And we all work together, but Sangha, she's a master's fellow. She also has done a lot of work, and Thomas, here, he did that co-immunoprecipitation assay, and it was, it's a very clean data you see, but to come to that clean data, it was a lot of time and energy involved. So that's about it. So that's, if you have any question.
I have a question. The sulfa transfer, the SALT2A1, it's a senescence marker protein, but I know you started out with the
Gene regulation.
But is that go up or down with the in senescence?
Up.
Up.
It's like your beta adrenergic receptor. It's like
that's
this.
right. Okay, so they're up. Amrina and I have just discovered a literature that you probably know very well, and I hesitate to mention too much about it because
No, I'm sure I don't know.
Because I probably don't understand it, and anything I do understand has just gone in at a grant that I hope is not wrong. But there's the actions of CEBP alpha as a function of age in liver, as I understand it, may be dictated not so much by CEBP alpha levels of expression, which
No,
may
it's the
not change with age,
alternate processing.
but
of the
rather,
messenger.
Participation in it binds as
Oh
part of
yeah,
a
that's the
complex.
Baylor, that
Yes,
guy.
right.
Yeah. Oh, he has it. That's a beautiful data.
That is beautiful.
Yeah.
That would indicate that the actions of CEBP alpha are being altered as a function of age on the basis of it being tied up in an age-dependent fashion in this binding complex by other proteins that are participating and causing it to shift into that binding complex.
Exactly.
So the actions that you're talking about that require CEBP alpha may actually be age-dependent in a way that would inhibit this kind of.
You know what? Actually,
reaction.
you gave me a fantastic idea just now. I never thought about it, connecting the two. I knew about that data, that CBP alpha complex, that big complex, that
Very interesting.
that's very interesting. But now, I can actually tie that with this regulation to make a very interesting case,
Well, that's what
at
you're
least
proposing to do
speculative.
with the beta receptor in our.
Uh-huh.
in our mechanism.
Okay. But I'm so glad you brought it up. Sincerely, I didn't think of that connection.
Well, if we get
but-
hepatocytes from young and old animals, we can do all of this.
all of this,
perhaps, so.
and we can talk more about this. I mean, that's the whole idea about giving presentation, talking about data and collaboration.
Am I
Amrita
right?
and Amrita and Mike and my lab, we are doing.
It's called a Brahma. isn't
Yeah,
it?
yeah. It's actually chromatin remodeling factor.
Remodeling.
Yeah.
Yes, right.
I mean, complex. Hi. Yeah.
The use of D3, why vitamin D3?
Well, that's the one. D3 has one hydroxy group, right? Or it has
Well,
both.
that's different.
It has both. It's a one.
It's
Yeah,
like
it's a
125
125 dihydroxy. The one that we are using is 1,25 dihydroxy vitamin D. So it's a biologically active vitamin D.
That's not D3.
No, so we should
D3
write
D3
that.
is cholic acid.
Right.
And
So we
then,
should really write 1 comma the whole thing.
um, The other thing is the assays where you look at fold induction of luciferase and you're looking at in some cases 7-time fold increase. I don't think anybody would argue much about that, but when you start getting down to two-fold activations or even 3, at least in the field of fatty acid activations of the PPARs, those kinds of activations are generally looked at
Yeah.
without a lot of pizzazz. I mean, because when you really get activations, you get 10-fold,
Right.
20-fold.
Yeah, no, I know exactly what you're getting at, and let me answer that because I have dealt with that, thought about that. I have lost priority of publishing a paper because of that.
Sorry.
So
No,
yeah,
I'm
no.
asking
First
for
of
my
all,
own-
this
for
is
my
no,
own,
yeah, no, I know.
education.
First of all, this is why any regulation you show in transfection, in transient transfection, you really have to show that in vivo also. Okay, as a matter of fact, I could not publish a paper. This vitamin D receptor regulation, the initial paper was in the mouse gene and mouse promoter. And originally we sent it to JBC. And the only comment came back, you haven't shown it in the animal. Okay, so, and we didn't want to wait because that's how we lost priority with the PXR regulation,
And they wanted
Yeah,
to show it in the animal?
yeah. So we just went ahead
If
and
you had shown it in the animal, then you didn't show it in something else.
well, whatever, but so now I have the animal data actually. Yeah, so that's first of all one thing, because of that limitation and depending on the condition of the cell, because sometimes what happens when you work with, that's why sometimes it's better to work with primary hepatocytes, but there is a problem with primary hepatocytes. Whenever you culture the cells, even in primary cells, many of the co-activators and co-regulators, they disappear within 10 hours, within 5 hours, all depends, So the thing is that when you don't have the whole complex there, you're not going to get the maximal optimal efficiency, okay? So the thing is that I lost a paper to a National Academy member, so what can I say, on PXR regulation of SALT2A. because I was looking at the lithocholic acid dependent. PXR is also very minimally activated by LCA, but much better by BDR. And in a transient transfection system, I was seeing like 1.5. If I push it under very optimal condition, 1.8 fold. I said, I cannot publish a paper with 1.48 fold induction because it was in the transient transfection system. Okay, so, but what happened is that Ron Evans at Salk Institute, who actually is a discoverer of PXR, he had the PXR knockout mice. So he published the paper in PNAS showing 1.5 fold induction in the transient transfection system. However, you have the knockout model to demonstrate very clearly the effect of that regulation himself. So anyway, no, but so what, I'm just giving an example. Therefore, this is something, so I'm very happy if I get, even so I have now with, by hard experience, I've now learned even 1.8 fold induction, if you see it again and again, it's nothing to sneer at, which I myself did at our own data, and we didn't publish it, but now I realize I could have published it. I mean, people in the field understand, but you do a lot of control studies. For example, let's say you have the promoter and luciferase, okay? You have the natural promoter and then luciferase. Then you add a control where you have luciferase without promoter to show that 1.5 or 1.8, and I don't publish a paper, I don't think I will ever do, without animal experiment to back me up, a 1.5 fold stimulation. But the point is that if you are going to show even a 2-fold stimulation, all these data that I show, we have the negative control where the vector alone, where it has the luciferase, does not get induced.